Hello and welcome back to the Lab 8 experiment EE542 Fall 2018 under Professor Yang Cho. In this lab, we are going to talk about AWS Elastic MapReduce Spark programming. This part has two parts. The first part is to try the sample program and the sample data provided to pattern match with the sample. And the second part is to sample and search for specific characteristics of the human genome data. This is also a team project, and the team members are as shown below. The main objective is to pattern match the DNA samples provided by the genomic data that we download from AMTDB, and we are using specific services by AWS, which one is EMR, Elastic MapReduce, which is used as a dimension reduction platform, which also uses Hadoop distributed file system to have multiple nodes performing processing information on the sample, and Apache Spark, which is the framework which is used by the Apache Open Source Foundation. And we will be using AWS Bucket for having our program data, and so that our EMR clusters can access them. Coming to EMR, it's one of the services provided by Amazon Web Services, and the cluster is made up of three kinds of nodes: a master node, a core node, and a task node. The master node takes control over all the slave nodes where the slave nodes actually perform the data processing. So this is provided by a Hadoop framework, which is of course cost effective since it's on cloud, and it securely manages analysis, indexing, which can also be used for machine learning, scientific simulation, and of course bioinformatics, which what we are trying to achieve in this lab. Now there are a few steps which are involved in creating Elastic MapReduce. The first one is start the processes, and then once the processing begins, it runs, and it is kept in a pending state. All the slaves are kept in a pending state until the processing is stopped. But in case they fail, as shown in the second image, in case they fail while they are processing, all the other future computing is stopped, and then it ends the process. HDFS is one of the main focus of our lab experiment. Hadoop distributed file system. It uses a master-slave kind of an architecture, which has, as explained in EMR, has a master node and other slave nodes, which duplicates all the data so that you can do parallel processing, and everything is a string operation. So it creates multiple data blocks and distribute them across. It's just like distributed computing, but in cloud, done everything through software. This is very suitable for large data sets so that we can divide them, parallelly process them, and of course each of these data have to be independent and identically distributed. Into Apache Spark. It is by the Apache Foundation. It's an open source platform which has a lot of features, which has SQL compliance, which is really useful for large-scale data processing, streaming, in-memory processing, task visualization, and immutable data. Using Spark, we are performing this lab experiment on EMR. So Spark is one of the main frameworks that is being used.
So part 1 of the lab 8 requires to look for a string pattern in a Spark program, and we're going to do this by creating a EMR, which is Elastic MapReduce cluster on Amazon. So let's start by creating the EMR cluster on Amazon. So basically in an Amazon EMR, there's a cluster, and a cluster is a collection of EC2 instances. Amazon EC2 is Elastic Compute Cloud instances, and each instance is called as a node. And the important aspect of this node is that it has a role. and every node has a different type. So before we create an EMR cluster, there's a few steps that we should follow. We should first create a S3 bucket, Amazon S3 bucket, which is going to store the output that we're going to get by running that sample code. And also, we should create an EC2 key. We need to create this key so that it stays secure. So here I am in my Amazon console. First, I'm going to create my EC2 key. So I have 0 key pair as of now. Create a key pair. I'm going to give it some name, say my key pair. Create. So I need to save this key at a very secure location. So this is going to be a .pem. It is saved. Now I'll go into my services, S3, and I'm going to create a bucket. I'm going to give it a bucket name, which is going to be, say, Phoenix. Create. Okay. Let me just give it EK. So I'm keeping my bucket as private, so I can only download the data that I get, and nobody else can view it with a link. So now that I've created a bucket, let me go inside it, and I'm going to create a folder named as input. And in this input, if I open it, I'm going to store my string data. So this here, my string data, I have downloaded it, and it consists of a huge data from which I need to search a particular ID. I'll hit upload, add file, and here's my string data. Next. And it's uploaded. Now if I go back, I want to also upload my pattern. So this is the code, this is a Python code which runs on the Spark application by Spark. And I'm going to, this is the sample code, and what this does is, let us check what this code does. All right, so what this code basically does is that we have a, so instead of this, let us open the assignment to find the exact pattern that we need to match, which is this one here. I'm just going to copy and paste it inside my code. So this is the pattern which I need to find in that string data. Pattern search, in short. All right, so this is 14. So what is it going to do is that it's going to go inside the string data, and it is going to match this pattern, and it is going to search that each pattern needs to have about 14 characters. And as we see here, this has 14 characters, which is why we have specifically mentioned as 14. And that is exactly what this code is going to do. So I have uploaded this code, but I've made some changes. So let me save it. And I'm going to go back, remove this because I made a change. Actions and delete. Uploading it again. So this is the improvised one. All right, so it's in this bucket. So now I've created a key, EC2 key, and I've also created in S3 bucket, wherein I have my code and I also have the data. So now let me go and create an EMR cluster. All right, so I'm going to hit create, give a cluster name. I give it as EMR cluster, then my S3 folder. So it is this one, and this has my bucket, which is Phoenix EK, has all the data. So it has the paths that it needs to go into in case you have multiple buckets. Select. Now, the specifications in this assignment specifies that we need to keep this as a Spark application, which is why the application which I need is Spark. Then I'm going to click m4.large instances. So what this does is it adds a default of 32 GB GP2 EBS volume per instance. And I've created 3 instances as mentioned in my assignment. 3 instances. My key is my key pair. Create a cluster. So this is in one step. The first step, which is the starting step, it is making all the parameters of the EMR cluster ready. Let us look at the presentation. So in this part 1, we have just created an EMI cluster by creating an EC2 pair and by also creating an S3 bucket. All right, so this is exactly what we did. We had a S3, we created a bucket, we kept the pattern search program into it. We also created a new folder called as input, and inside it we kept the string data. And in this same folder, we want the number of string IDs which match. So we when we execute into the terminal, we will make sure that we write the command such that it creates an output inside our S3 bucket. So this is still in the starting mode. There are 4 modes, which are starting, running, waiting, and terminate mode. So we need to go into the waiting mode in order to start with the execution of the terminal command. Fast forwarding it. I have my EMR cluster ready. So let me open it. So I need to SSH from my terminal. And I need to do this to establish a connection with my master node, which is created on the EMR cluster. And I need to replace the key with the location and the file name of the private key, which is used to launch the cluster. So by using this command inside my terminal, I will be able to do that. All right, so my EMI cluster has been launched. And so now, according to my assignment, I need to run this command. And by running this command, I'm going to get inside my EMI cluster, which I already am in. And what's going to happen is that I will be able to get the string ID. And this string ID is matching for the number, for the string pattern that we had inserted inside the pattern search.py, which is the Spark program, basically. So this is being run at this moment. It may take some time. Till that time, let us look at the presentation. So in the command line, we typed in the command that was given to us by the Amazon. And by doing this, what we're actually going inside the EMI cluster, and then what we need to do is execute it. And by executing it, I ran this Spark command. So I should get something as succeeded, which should be the final status, which means that my output folder has been created, and inside my output folder, I will get a string ID. All right, so it says that the final status is succeeded. So let me go back into my EMR console. I'm gonna hit close. Let me go into my S3 storage, and that is because my output is going to be inside this. So it shows that it was succeeded. This is my part. So now if I had made my bucket as a public one, if I access this link, I would definitely get a string ID. But because it is private, I need to download it. Let me go into the downloads. And here you go. I've downloaded many times to check if I was correct or not. So it seems like just one string ID matched the pattern that we had inserted. And this was basically to get which is this one, 6RZH.
Human genome data. Now we all know that we're all made up of proteins, small protein samples, which are called DNA, deoxyribonucleic acid, which has data about our ancestry, our characteristics, and whatnot. So it's a sequence of nucleic acids, which has 23 chromosome pairs, and it is the smallest DNA molecule found within individual mitochondria in our cells. Genomic study leads to advance in diagnostics and treatment of diseases. So our future scope with this is to sample the genomic data and then find the traces of cancerous cells, and then try to improve the prediction of cancer, which is currently at 30% success rate. We are downloading the data from EMTDB, which has around 1,136 samples, which are spread across 3 continents, a lot of countries, right, and different cultures and different regions. So this sample is from Homo sapiens, taken from the mitochondria of Homo sapiens, and is provided by the National Center of Biotechnology Information by National Library of Medicine. A lot of people use this data to train their models.
Now moving on to part 2. Over here we are given the sample code and we have to modify to look for persons names that have the ND4 gene in the provided DNA sequence data, and the output should be in alphabetical order as shown below. The first step is to download the DNA sequence of 1131 people from AMTDB as given over here in the link. And we have to write a Spark program which will look for the DNA which matches the sequence, and on opening this link it will redirect us to the sequence of Homo sapiens mitochondrion, and this is the link which we are looking for. The steps are as given. First step is creating a EMR cluster. I'm going to use the same EMR cluster which I used for the part 1, as the configurations are exactly the same, where the application would be Spark. There would be 3 M4 large instances and I have to create a bucket. Now I will create a new bucket for this experiment, as the files which are going to be uploaded are going to be different. As it shows over here, I'll name the bucket as DNA cluster. After creating the bucket, I'm going to upload the code as well as the data containing the sample DNA sequence of 1131 people. mtdb records.fasta contains the data of 1131 people, and dnasearch.py is the Python code for Spark application. I will show it to you in a while. So both the files have been uploaded in the bucket. Now I will have to SSH into my cluster. For that, this is the command as shown over here. After this, I need to run this command, Spark submit command. Just to be sure, I will check over here if the naming is perfect. DNA cluster 1, DTB records.first, DNA search.py. Okay. Now going to the code. Over here, as this is the pattern I'm looking for in the gene of 1131 people, I have pasted it over here, and on opening the list of the records, we get to know that the ID of the person is almost as long as 6 or 7. So over here I have modified the length of line to be 6 or 7 so that it can show the ID of all the people where match has been found. Now as it shows, the final status has succeeded, and output should be created in the DNA cluster bucket named DNA output. We can check it by going over here on refreshing the page. Here is the DNA output, and as it shows, it is success. And on downloading the part, I can find it in my downloads, and as it shows all the IDs of the people whose DNA has been matched, just as we are asked to find. As it shows, all the IDs are in an alphabetical order. So the cluster has been created as shown. The bucket was created with the DNA output, the data records, and the code. And here is the Spark program. These were the command line arguments which were passed first to SSH into the EMR cluster and then to run the Spark command. And this was the output which we got as I showed you. So that concludes the part 2.
Once again, thank you for viewing the lab. Please like, share, and comment for feedback. We'll always welcome feedback so that we can improve. Thank you, and see you in the next lab experiment.